Hello and welcome to Nikolay's genetics lessons, and today's video is going to be about gel electrophoresis techniques. And here's the first question: the rate at which DNA migrates through the gel determined by, and we have to choose one of the answers. But first, a little bit of theory. Imagine that this is top view of the gel matrix, and now let's imagine that we load our gel with different size of the DNA. So let's say this is single-stranded DNA. One would be long and would move through the matrix. Another one would be shorter, and of course it would be easier to move for the shorter version of the single-stranded DNA than if we compare with the long version of the single-stranded DNA. If we'll take a look at this picture, we would see that double-stranded DNA is slightly negatively charged. Take a look. Negative charge here, 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 here. So we can find negative charge on the oxygen, which we can find on the backbone of the double-stranded DNA, or it can be single-stranded DNA. Our slightly negatively charged DNA moves away from the negative charge and towards the positive charge, because DNA itself is slightly negatively charged, so it's going to move in this direction. Now let's check our answers. A molecular size of the DNA, and as you see, the smaller molecule, the easier it's going to move through the gel matrix. And eventually these fragments of the DNA which make the same size DNA would make a band on the gel. So this is correct variant of the answer, but let's check the other answers as well. The applied voltage, of course, the higher voltage, the faster our DNA would move through the gel. So this is also correct variant of the answer. Applied voltage does affect the rate at which DNA migrates through the gel. So next, the electronegativity of the DNA. It also looks like correct answer, but actually, as I said, DNA on the overall has negative charge, but it's directly proportional to the size of the DNA. So smaller fragments of the DNA would have smaller number of negatively charged oxygens, and the longer the DNA, we can find more such atoms. So negative charge of the DNA is the reason why DNA moves through the gel, but doesn't affect the rate at which DNA migrates through the gel. Now we can choose the answer, and this is going to be variant D, both A and B. The second question: why do scientists load DNA known sizes into the Agarose gel? Take a look at this picture. So what we see on the left we call ladder, which consists of the DNA, it can be RNA or proteins, of the known size. Usually biotech companies that sell such ladders produce them by cutting DNA of the known strains of the bacteria with known DNAs, which cut them to fragments of the known size. And later, other scientists or laboratory technicians, when they run unknown DNA, they would run along with this DNA ladder, and they would use this DNA ladder as a reference in order to find the size of the unknown samples. If we see that on the ladder this band is 1500 bases, then we can suggest that the size of this unknown sample is going to be 1.6 kilobases or 1600 bases. And for example, unknown sample, which is here, would be about 2.9 kilobases or 2900 bases. Now let's read the answers and let's choose the correct answer. It makes it easier to determine size of the unknowns using comparison techniques. And this is correct answer, but let's check the rest. For example, to fill in all the slots of the gel so you can run it, it's not the correct variant. Next, C, to practice loading the DNA before you get to the important DNA, this is not true. And so you know how long the gel needs to run is also wrong answer. And this is all for today. Thank you for your attention. Please subscribe for my new videos that I post almost every day, and see you in the next video. Goodbye.